powerful, right? Okay, so now our goals for today is I'm going to teach you all I know about UniProt. And in my opinion, it is one of the best resources. It's actually a gold mine for retrieval of data. You can do powerful research using UniProt, right? So not because I am part of it or it's next door to me or anything like that, but it is actually pretty good in terms of the team. And of course, so is NCBI as well. And there are lots of other resources, right? So, but these are the two hubs that if you know these two hubs, if you know all the data that's there, you can be busy for the next 20, 30 years because there's so much data there, right? And so we will do some customized searches. We'll do some blast searches and then find orthologs. I'll explain to you as we do this, multiple sequence alignment, and actually just a small phylogenetic trees. We're not doing anything, any rigorous evolutionary analysis. And of course, I promised you yesterday that we'll actually do genome-wide association studies catalog. We can actually look at it. Hopefully we'll have the time to do it. I think we will. Okay, so how to make best use of information stored in any database. What is the best? So I'm telling you, standing here and telling you that NCBI is one of the best. It has so many different databases. It has lots and lots of information. It has lots and lots of data. And UniProt also has lots and lots of information, right? So how can you make best use of all the information that is there in UniProt and NCBI? How can you make best use of it? Any database for that matter. How do you make best use of any database in a databases? By learning how to search them efficiently, right? Sometimes the problem is databases are not created, they are so confusing. There are too many buttons out there. You just don't know how to search them. So you just do it. Yeah, how to navigate. That's correct, Rahman. I think you've said it really nicely. How to navigate a database is actually the biggest challenge for all of us. And if you learn how to navigate something, that's it, right? So you know how to navigate, you're there. You will be a good GPS system, and then you will know exactly how to get information and where to get information from. And I'm standing here and telling you that UniProt, you can do so much with UniProt, and that resource itself is sufficient for you to make discoveries, right? Because there is so much information there but you just have to know how to pull data from, right? So from UniProt database, of course, you can do millions of different things. How many genes are there in a human genome? How many genes? And how big is my protein? And what is my protein? What diseases is it implicated in? And all kinds of stuff, right? So we're going to be doing some of these or all of these actually. So now is the time for us to actually get to hands-on. So if you all can open your browsers and go to uniprot.org. I think I'm going to paste that in your chat box. Okay, so I'll just give you a second for you to actually get to UniProt. Okay, is everyone here? Right? Hopefully you're all here. So, so this is UniProt, and it brings me all wonderful memories of my time at UniProt when I was doing curation and adding lines to some of these entries that's here, right? So this is UniProt symbol, and so it has a blast. You can do alignment. You know, you can retrieve and map different things, which we will go to if I have some time, and then we can do peptide search. And of course, you know, this is another language, I believe, that you can actually pull up things if you know programming. Now, UniProt, the screen is, okay, so I think, screen is frozen.
Yes, Dr. Sona, we can only see the presentation.
What's that? Oh.
We can only see the presentation. We don't see your screen.
Oh, really? What happened? Okay, let's see. Can you see it now?
Yeah, now we can see it. Yeah.
Sorry about that. I have no idea what happened. Yes, good, good, fantastic. Back in business. I think maybe I just moved the PowerPoint and I have several windows. So maybe I'm sorry about that. I apologize. Okay, so now we are all back in business, right? Um, so what I was saying was, I'll repeat myself. So we UniProt, and you have, Blast, you have a tool for alignment. You have a tool that if you have lots of IDs, then you can actually retrieve everything in one shot. You can do a small peptide search, and all of those things, right? So this is all. And then this is the reference clusters I told you about, where you cluster proteins that are similar at different cutoffs. And this is an archive, right? So this is actually very, very, very important, because say that I am working on a sequence today, and I'm working on that sequence from UniProt. I downloaded the sequence, and then I'm working on it. I'm doing research, and I'm just ready for publishing my work. And then I go back to UniProt to check that sequence, and it's actually not there. And why is it not there? Because the primary sequence, the author who deposited that may have actually withdrawn the sequence, or UniProt actually, said that there is a problem. It's just a fragment, or there was another sequence that was of better quality that came in, and so that they removed it. So all these issues, all the scientists was facing. And so what we decided to do was, well, we'll say every single sequence that comes in, we're going to store it, so that a scientist who wants to look at a sequence that they worked on 10 years ago, they should still be able to get to the same sequence. So we created this archive. And then this is proteomes. This is where you get all the completed proteomes that you have. And then we have all of these supporting data. We have lots of genes that we talked about that are implicated in diseases. And then this is actually a very, very integral part of UniProt. It's called the cross-reference databases that I'm going to talk to you about, right? So in my opinion, UniProt is a one-stop shop to do everything, actually. Although it is a protein database, but you can also get DNA, and you can get lots of information based on the cross-reference databases. This is actually, I will explain this to you a little bit later, right? Subcellular localization and keywords and all of those things. The most important thing that I actually want to talk to you about is UniProt knowledge base. If you were my students taking my exam, these are questions that will come on my exam, right? So UniProt knowledge base has two different databases, right? One is called the SwissProt, and one is called the Tremble. So what do you think is the difference between these two? It already tells you here, right? SwissProt is manually curated, and Tremble, remember I showed you where from the truckload of these sequences, where we just translate it by a tool, translating tool, and it gets the tier where it is just automatically generated, right? So there is a difference in my view. I'm not sure if everyone else agrees with me, but this is something that I teach my students, and that's what we follow here in UniProt. Is there is a difference between annotation, and there's a difference between annotation, and there's a difference between curation. So manually annotated is curation. This process is called curation. Anytime you see something is curated, you know that some human being has actually looked at it and actually added data. So within UniProt KB, what we have done is we give it a golden star. So this is how you know whether a sequence is actually reliable or not. If a sequence is reliable, then you see this golden star. It gets a golden star, which means someone like me has actually looked at the sequence, has actually added information to that sequence, and then you can actually trust that sequence, right? Anything that just says Tremble, I wouldn't trust it, because it's actually just automatically generated. So maybe the open reading frames are wrong. Maybe it's shifted, frame shifts, and all of those things. So I would just rely and trust anything that has a golden star, right? I still remember the meeting we all had to figure out what can we do to tell the community that something is trustable or not. Okay, is that clear? Okay. So now, of course, you have lots of other things, and we have lots of information about COVID and everything else as well. So now what I want to do is, we can actually just, like Google, right? We can just type in sickle cell anemia, right? Okay, I'm just going to click, and everyone should see something like this, right? So why is it pulling up? So it has pulled up how many total? It has pulled up a total of, it's 10 human proteins, right? It's pulled up only 10 human proteins. And then out of which, how many of these sequences can you trust? The ones that actually are reviewed and has a golden star is now 6. So if I click on that, it's only all the reviewed sequences. You will see only 6 sequences here, right? So now what is, I want to pull up, I want to look at hemoglobin, right? That's correct, Karina. So I want to pull up hemoglobin. Now it is pulling up pyruvate kinase. This is what I want, right? I'm looking for hemoglobin because I want to spend the hour looking at hemoglobin sequences. So why is it pulling up pyruvate kinase and all this creepule-like factor? Then of course, this is again hemoglobin subunit, gamma subunit, right? And then it is pulling up some other receptor and all of those things, right? So why is it pulling up all of those? Because I want only hemoglobin, right? But then how does the search engine know that my mind is looking for hemoglobin, right? But what it is doing is it's like Google search. Anytime it finds some, I'm sure there is sickle cell anemia in any of these entries here. So it's pulling up all of these other entries because sickle cell anemia. So how do you make your search very specific? So how do I just get only human hemoglobin beta chain that causes subunit beta, right? That's what I want. So how can I tell the search engine more precisely that I want only hemoglobin beta chain, right? So these are all something that's are not very trivial and people don't talk about it, right? How would I have gotten only one entry, one result to my search? And that happens to be the result that I want, which is hemoglobin. Any guesses? How can I make this search more specific? Keywords, yes. That is actually a keyword. That is actually very good. So a keyword could be, of course, we have to make sure that UniProt has actually used that. Okay, so keyword of sickle cell anemia is a keyword, and that's probably why, Karina, you brought a good point, that it's actually pulling up all of these. Probably that is there in their keywords. Now using exactly. So Parna actually has the answer to the question. You need to have the exact entry or the gene name. And I saw protein of interest. So if I just type in hemoglobin, maybe of course it's going to bring up a lot of hemoglobins, right? So the way to make it very, very specific is actually to understand that each protein, like you and me have a, so of course we are for Indians, the same name can exist a million times within the country, but of course we all have something different in each, right? So we all have a name or something like that that's unique. So that is a unique identifier. So most of the databases have an identifier, which this is actually called an accession number. And this is actually the entry name. It gives you the gene name, and also it gives you which organism it's coming from. And this is actually the gene name. It tells you which organism it's coming from. And actually, Pihu has said, can you just search hemo? Yes, as Pihu, I actually said, we can actually try doing that. If you just search hemoglobin, you will get all the hemoglobins. But again, you'll also get entries where hemoglobin is mentioned in somewhere in that entry, right? So it's still, it is closer to what we want, but it may not be exactly, right? Yeah. So now hemoglobin, right? So this is what I want. So I'm just going to click here. So click on that. And this is what is called a protein entry. How will you know, be able to know the entry ID? Exactly. That is actually a fantastic question, Linnea. So how would we know? We don't know. So this is exactly where we will start off, and we'll actually look at the entries and see which one is actually correct. So most of the time, if you're reading a literature or a publication, they probably, and if you're interested in something, they probably will give you the entry number or accession number. So you can go from the publication, or you just say hemoglobin human. Yes, you're right. You may not be able to know the entry number off the top of your head, right? So you will find the entry number based on, if you know the gene name. and then you can just type in the gene name, or you get the entry number or accession from a literature or a publication that you're reading. For most of the time, we will be doing what exactly I did, right? We just put in some keyword, and then you go through each of the entries, and then you probably look at it, right? So in literature, if 2 or 3 labs research I'm reading, then how do we decide? But they should have the same accession number, Parimala. As long as they come to UniProt, even if it is 100 labs that are working on the same thing, they should actually have the same accession number if they're referencing UniProt. So it is actually non-redundant, right? Each accession is only unique. It's a unique accession, right? So you won't have that kind of issues. So now hemoglobin. So I talked to you about curation, right? So you will appreciate people like me who do curation, you will appreciate how much work we have to go through, right? So we actually have to, this is all, so it gives you, should there be a master list of accession numbers? Yes, actually there is a master list of accession numbers. And Linnaeus, I think you've brought me to something that I was actually not planning to talk about today, but remind me a little later, Linnaeus, if you don't mind, if you have time, just put it in the text box, say ID mapping, and I'm going to tell you, yes, where that list is. That's actually a very, very, very good question, right? We will get to that. There is a master list, and I'll tell you how to use that master list, right? So now, so hemoglobin, this is the name, we have the gene name, and this is, can we trust this entry? This is called a protein entry. Do you think we can trust everything that is there in this entry? Yes. How do you know that we can trust this? The answer is correct. Yes, it has a golden star, right? And yes, absolutely, it is Swiss Prot. It is curated because it has a golden star, and it says it is reviewed. That means, and it also says that this protein is actually, we actually incorporated this only about maybe several years ago, because people started saying, can we trust whether this is actually annotation score, correct, Kylie? This is something that we added new to the database, because the scientists around the world were saying, how do you know that this is actually an express protein? How do you know this is actually a real gene in the humans, right? So we started adding all of this levels of experimental evidence at the protein level. Then this section gives you function. And now what we have actually added is every line that we add here is actually supported by a publication. Remember, the primary data, we are not the primary, this is not our data, right? It is actually someone else's data who has deposited, right? So we are just curating, this is just a secondary aspect. We are just adding information to the primary sequence that they deposited in databases. Every line function, there is a publication that you can go through, and you can say, if they say this is involved in oxygen transport, how do I know that it's involved in oxygen transport? Because I've read this paper and it actually supports that, this supports this particular function. So everything is actually curated, and everything, it's a lot of work, as you can imagine. There are days that I would hate my job, right? I hated my job both at NCBI and UniProt, because all you sit and do is review literature. And then you add all of these things, right? So all of this you can, and of course, if there are some things that are actually bothersome for us, then we actually add that as a caution. And so now, then we have here, you can actually have all of these things. So in this particular entry of hemoglobin, you see tick marks in every part of this entry, right? So which means you have information, you have data that's there that is available for every one of these things, right? So you can get the functions, and then if you want to understand subcellular localization, you just click there, which means where is this protein actually expressed in the cell. So you can see whether it is extracellular exosomes, and it is actually a cytosolic protein. And you can get all the information about subcellular localization. This actually becomes very, very important for research purposes, right? And then if you want to understand what are the different diseases that this particular gene is involved in. Do you think every single gene is involved only in one disease, or genes can actually be involved, can cause multiple diseases? What do you think? Do you think hemoglobin causes only sickle cell anemia, or it can actually cause other diseases? Yes, of course. Correct, Jeffrey. So I think we all know that, right? A single gene actually can cause more than one disease, which is interesting, because then you know that all those diseases have something in common, which is the gene, right? The same gene can cause multiple diseases. And if you draw a network of genes and also um, all the diseases that they cause, you can draw a diseaseome. That's what I actually work on. So I have a question here. Can you please guide me through the last part? I got logged off because of the internet connection. So all I'm doing, Suparna, is actually just walking everyone through every aspect of this database. That's all. So I don't think you missed much. I'm just here, subcellular localization of where the protein is actually expressed in, right? So now, where is it found, right? And then you can see which part, where is it tissue specificity, which tissue is hemoglobin specific to, it's specific to the red blood cells. So pretty much every click you get is actually very, very important. So now let's click pathology and biotechnology. A section, my favorite section in the UniProt, and in this used to be called involved in a disease when I was a curator at UniProt, but now they've changed it to pathology and biotechnology. So it's involved in a disease. So you can see that this particular gene is involved in Heinz body. It is involved in anemias, all sickle cell diseases, beta thalassemia. It's involved in sickle cell anemia. It is actually involved in dominant inclusion body type. So if you go to UniProt and if you click on the pathology and biotechnology section, it will tell you all the diseases that this particular gene is actually involved in. And it will give you all the publications that is relevant if you're involved in Heinz body anemias and all of those things. So this is actually a very, very important part of UniProt. The other important part of UniProt is it actually gives you all the single nucleotide polymorphisms actually that exists, that is causing, that corresponds to these two diseases, right? So these are all the different variants. Both some of them are experimentally validated and some of them are not experimentally validated. So this one tells you that it confers severe malaria, and if you want to see where is that information coming from, then you know that it is coming from, you can read these publications and it's coming from these publications, right? So this is actually very, very important. And of course, it is actually connected to this. If you want to get more information, it is connected to a database at NCBI. I'm not going to click right now. We are going to go there. We are also going to go here, and I'll show you all this. So this is actually a lot of information. Now, this is sickle cell anemia. You already know what causes sickle cell anemia, and look at that. It is actually, it is telling you here that the natural variant is glutamic acid to valine, and this is all the publications. So, okay, so the question, that is very good question, Linnea. So yeah, so any one of this can actually cause. All this means is actually, of course, you have to read all of these publications in detail to see what they're actually saying. What it is saying here is saying that this SNP from L to P is actually observed in people with beta thalassemia, right? And this is actually, and it corresponds to this particular variant. Now, so if you actually, so it does, do people with this actually need to have all of these 4 different polymorphisms? Is it something that actually is your question? Actually, no, and not necessarily. But honestly, I've not read all of these publications, Linnea. So maybe you can actually, you know, go through these publications to see. But all it is saying is they got that L to P implicated in this particular disease based on the publications that they're giving here. That's all this means. If I know of a disease I'm interested in, can I just, Kylie, you're going ahead of me. We're just going to do that in a second. Okay, I'm going to show you how to do that, right? So yes, you can actually search for that. And I'm going to show you a little bit of advanced searches of how to get information, right? I'm going to do that. So now that's what it means. And now, so keywords, I don't remember now. My memory is, someone actually talked about keywords early on, right? You can go through if you know keywords. So you have, these are the major keywords, which is actually, you know what? This is actually curated keywords. So everything that I'm talking so far, all of this is actually curated information, right? Curated means that someone like me has actually read literature and adding information. Now you have many proteins have actually glycosylation. That means a sugar is added, right? So this is N-linked glycosylation. And now all of this actually has a reference, right? Now, again, you can trust it or not because there's only one publication that actually tells, but then there are a lot more publications that you can go here and read, right? So this is, tells you if you want to see which position of the protein is actually glycosylated. So you can go through all of these amino acid modifications, and then you can go through interactions. You can actually go through interactions. Now I want to get to, so cross references, right? Now, what are cross references? That is something that is actually very important. So UniProt has parts that is curated, and part of the information comes from other databases. So all the information about hemoglobin beta chain from that is relevant to this particular gene, coming from all of the other databases, is called cross references, right? So you can get information about hemoglobin beta chain from all of these other databases, which is not part of UniProt. That's why they're called cross references, which means UniProt cross references all of these information from other databases. So here you have cross references. If you click on cross references, right, you have lots of, so if you want to get the DNA sequence, gene sequence of hemoglobin beta chain, you can go to, I told you, right, these are the 3 different resources where the sequences are stored. So you can actually go through 3 differences. You can just click on these things, and then you can actually go to, yes, actually, you know what, Parimala, these that's actually an excellent question. So these cross references are not curated by us, but what we do is there is a committee on cross references. Every database that has to, so many people come to us and says, can you please link our databases from UniProt? Because UniProt is a very popular resource, right? But no, we don't link anything and everything to UniProt and entry. There is a review process where people actually go and look at these databases to see if these databases are reliable and if these databases are actually curated those information. So we don't curate them. We just actually have a committee that looks at these resources to make sure that they're reliable resources, right? So I hope that answers your question. Then again, it is actually PDB. This is actually a very important thing for you to understand. And we'll be doing this tomorrow. All this means is that the structure of hemoglobin, does UniProt have step-by-step manual on how to navigate these? Yes, there are lots of actually manuals and tutorials that are available. And I think it's at the very end. Yes, there are lots of tutorials available for many aspects of UniProt. And UniProt help desk is amazing. They just actually respond to you overnight with any of your questions. If they don't, just let me know, because I can just pick up the phone and call them because I know all of them, right? So you can actually, they're very receptive to those things. Now this section is structure. So if, for example, next week Ilya tells you go and pick up, pull up a structure. In fact, we'll be doing that tomorrow. So pull up a structure of hemoglobin, or for your projects later on in this course, right? You will actually, for the course, if you actually want to pick up, I think, I don't remember, I think it was Jeffrey who asked me yesterday, how do you pick up a research question, or how do you pick up a research protein? You can actually, after today's, all of the hands-on we do, you will know how to pick up something that's very interesting, right? So this is the section where you will go and see if the protein of interest actually has a 3-dimensional structure available, right? So now look at how many people in the world are actually working on hemoglobin. Oh my goodness, I can't even count the number of structures that there is for hemoglobin. It's completely beaten to death, right? So many people are working on hemoglobin. So many people have solved the structures of hemoglobin. And you can actually pick any one of these, and this tells you what region of the protein sequence that the structure is of. And this is the structure. This is actually a very important part of the UniProt. And then it goes on and on and on, right? So this is all that, whether there are any, whether this gene is a target for any of these drugs. So these are all the drugs. This is another database. It's an amazing database called Drug Bank. So you can actually go and, so is there a nomenclature for ID for structures? Parimala, all I can tell you, yes, there is a nomenclature. It's always a 4-letter code. It always starts with a number, and then it's a combination of numbers and letters. Now, we had to actually, yes, all accession numbers, honestly, actually has a pattern. And that's another lecture to tell you how accession numbers, we came up of accession numbers for UniProt and how we came up with, even PDB came up with this accession number. So it's always a 4-letter code for PDB, and everything has a pattern. You're right. That's a beautiful observation, right? So, and then you have all of these different databases. DisGeneT is another database that I'm really liking a lot. And then you have pathways, if you want to look at which pathway something belongs to. So it is actually all of these miscellaneous, you can go through every single one of these and learn from all of those different things. CDD, this is where I worked when I was at NCBI. It's a structural database again. And then PFAM is another domain database, and it tells you what domain does your protein contain, right? So if you click here, each of this click is going to take you somewhere else, right? I'm not going to do that there because we could get completely lost right now. I just want to focus on this. And now it tells you which version it is. And one of the biggest nightmare, I'm happy I'm not running this resource, is how do you keep it updated, and how do you, if there is new information, how do you recurate something? That's all for another day, right? So there's so much of information, medical and all of those things. So this is all pretty much cross references. It tells you which chromosome it belongs to. It belongs to chromosome 11 and all of those things, right? So in a nutshell, this is called a protein record that contains information about just one protein, right? This is all about hemoglobin beta chain, right? It's all about one information, and it all contains about one information. Now, I think one of you asked me, how do I find something? So the next thing is I'm going to show you how to search for something. Now, do you all remember that we actually played this game, or I actually asked you how many genes are there in the human genome? Remember I asked you that question? Do you all remember how many genes we have in the human genome? And how many? Yes, about 21,000. We can find out exactly. And then do you all remember how many genes that we found that were implicated in a disease, for example? Do you all remember that number? How many genes? Yes, Moeva, right on. My goodness, you guys are phenomenal, right? So let's do, I'm going to show you how to do that, right? So click on this advanced search, and you can do a lot of different searches, right? So I am going to ask you one question. I want to pull up all the human proteins that are curated, right? How many human proteins are there in the UniProt database that is actually curated? Now, if you know how to do searches and build the searches, you can find things in a click of a button, right? So let's do that. Oh, okay, let's come back to this now. So before that, right? So what I want human proteins, right? So you can actually use any one of these to build searches, right? So I am going to use, which one would you use for me to tell I want only human, right? So you will click on organism, right? And then I am just going to type in, and we have a very special name, right? We are all called, we have a very, very special name. Do you all know what we are called? Humans, of course, but what genus? Homo sapiens. Okay, that's correct. Homo. You start typing, and in a second it should pull up Homo sapiens, right? So it pulls up Homo sapiens, and this number next to this, it's called the taxonomy number that comes from NCBI. Each species actually has a taxonomy number, right? So you just click there. So what are we again doing? We are going to pull up all the human proteins that are actually curated, right? How many proteins are there in the human genome? Let's see whether my number was actually correct, right? And what I want is to say, okay, this is all human proteins. Let me move this out. And I am just going to say, just search, click search. Should be very quick. And you should see all of the human proteins, right? So how many is there? How many proteins is there? So I was right? We have 20,368 today, right? Let's see, hopefully it'll stop at 21 at some point in time, right? So this is all the proteins from the humans. Now, the next one I want to tell you is, okay, go back to the advanced search. Out of these 20,000 proteins, how many of them are actually experimentally validated to be involved in a disease? And we came up with the number around 4,000, right? Let me see if I was actually correct, right? So click on this button plus, that means we're just adding more to the query. And what I'm going to do is, do you know which of these keywords that we actually will use that will contain disease information? For example, which of these tabs that you think I just talked about that? Which exactly, pathology and biotechnology, right? And then you will click on disease here, right? And so what I want to know is if you want specific disease, I think one of you asked me that question, can we look for specific diseases? Here is where you would put your specific disease. Say you're interested in cancer, you're interested in, these are things that you're going to be doing for your activities, projects, right? You're going to be learning today and you're going to do things on your own, right? So pathology and biotechnology, I'm just going to press the search and look at this. I was correct, right? We were almost close. So we have about 4,454 of these proteins are actually involved in the disease, correct? So this is how then we can keep adding more and more to this, right? For example, out of all of these genes, how many of these are implicated? Do you have any specific disease that you guys want to look at? I know each one may have a different disease, but whoever is going to type the first disease, we will actually look at that particular disease from here and see how many genes are there for that particular disease that you are interested in. Diabetes, okay, type 1 diabetes, okay, so we will do that. Now, one thing that I want to say is, if you click here, right, columns, it's going to give you all kinds of information, right? So what you want is, I think I want to add, say, for example, structural information. I want to see, sure, absolutely. Please go ahead. So if I want to add a structure, right? So if I want to see if there is a structure available, then I'm going to click here. Anything that you want, additional information that you want. Say that I want polymorphisms. Where is that? Okay. So I want to see if there are any SNPs for these diseases or any of these entries, right? So I'm going to click on DB SNP. So you can keep adding. And I want to see what pathways that these genes belong to. There are several different pathways that are actually, cross-linked here. So I can actually create a pathway link, say reactome is something that I really like. So then you click on save here, right? And then you will start seeing all headings here, right? You will start seeing everything that I'm adding. And the wonderful part of all of this is you can actually download the entire thing as an Excel sheet, and then you can work on it. That's what I do. I do all my searches and download, just click and download. So now let's see. I think Karina wanted to look at type 1 diabetes, but of course, Karina, I don't know what this keyword that they've used, but then I'm just going to put in diabetes. Okay, that's okay with you. So what I'm going to do is, out of all of this, I want to find out how many of these genes are actually, if we know any information about diabetes, right? So I'm going to type in diabetes, right? I don't know if I'm spelling this correctly, I think. And then if you click, there you go. So you are having 143 out of these actually are in place. Somewhere diabetes is actually shown in them, right? And some of them may be because there is a keyword, or some of them actually may be because they have a diabetes somewhere else, right? So now what else do you want to add? Do you want to add anything more to this? For example, I want to see how many of these actually maybe have a three-dimensional structure. How about that, right? So click here. Now I want you to tell me how to do this, right? So you just click here, and how would I add? So tell me which keyword you would use to see if any of them actually have a structure. I may have already added that, but let's see. So which keyword would you actually use to see if any of these 143 entries actually have, yes, Michelle, correct, structure. And I want to say 3D structure available, because I just don't want a secondary structure, right? So 3D structure available. And now you click on search. Wow, 150% of those actually have a three-dimensional structure, which means we can actually do a lot with them, right? So fantastic, you guys are amazing, right? So Now, all of this is how you actually search. And as you know, right, we have limited amount of time. I want to still get to a few more things, right? So now we can keep adding other things. And if we have time at the end, then I will ask you, I will give you an activity that you can do while we can do an activity together if we have time at the very end, right? So right now, I just want to get to show you everything that we have. Now, is there anything else? So this is how you actually search for things and you actually look for things. And remember, always make sure you see that golden star so that you can trust everything else, right? So now this is amazing, right? One shot, you can download this and you can actually look at things, correct? Now what I want to do is, actually, I missed the audio for a few minutes. Can you please repeat after column disease involved? So all you do is, what we did, Parimala, is to actually go back and out of all the 143 we found for diabetes, we actually found everything that actually has a three-dimensional structure. So I went back and added three-dimensional structure there to the database so that you got 77 of them actually have structures, correct? So now you can download everything and look at all of those things. Now, if you are all, if any of you know programming or any of you know all of these languages, this is something, it's a binary thing. You can just, if you know how to do this, you can just type things in. I still have not learned it. I still go through the, the things and then do all of those things. So I know, it's amazing and you can actually look at ontologies and all of those things, correct? So now what I want to do is I'm going to take you a little bit touring to another two resources, right? So now I'm just going to get back to our hemoglobin example, which we can actually just do by going back. I think we can just go back, or you just type in HBB_human and you should get back to your, there we are. So is everyone back to hemoglobin? I want to show you some few other things. Yes. I'm just going to give you a second. Okay. So now if you are in hemoglobin, right? If you click on pathology and biotechnology, right? So I'm going to show you where else you can get more data from, right? So I'm going to focus on sickle cell anemia, right? Now, sickle cell anemia, it is only one variant here, right? And it is giving you, Ensemble is just another resource that is just amazing. That gives you a lot of data and lot of information, lots of population information, phylogenetic analysis and everything else. It's beyond the scope for this particular hands-on, but it's another whole new thing that we can do. So now let's click on what is called, let's first click on this, right? DB-SNP. And someone asked me that everything has actually an ID. Yes, anything. If you want to look at SNP, it has a very specific nomenclature. How do we learn what the exact mutation that causes the disease? Yes, what is the exact mutation that causes the disease? Of course, the best way, Kylie, is to actually read the literature. And I'm going to show you this resource here in a second where you can actually look at. And that is actually going to help you, right? Now, let's look at this. This is a database, right? Any time you have data, immediately the next thing to do is create a database for everything. So DB-SNP is result from which project do you think? DB-SNP was created from which of the projects that I talked to you about? Was it on Monday? Which we discussed a lot of different projects, correct? We discussed a human genome project. We discussed a lot of projects. Which project actually was the cause for creating this particular? Exactly, fantastic, HapMap. I think Parimala, I think you're actually, your texts are coming only to me. Maybe you can just text it to everyone, so everyone can actually see it. So if you click here, DB-SNP, and it takes you completely to a whole new world, right? It is taking you to NCBI. You know what? I'm just going to go back and I'm just going to say, open in a new tab so that I'm not going to lose this tab. I want to go back to hemoglobin, right? So now this is a whole new world. This is a new look and feel that DB-SNP is before it was. So it gives you all that information of what this particular SNP is. And and actually, Kylie, you can actually read all of this to figure out also all the different publications that are coming out, but I'm going to show you another. So these are all the different builds of the genome sequence. And I don't think I'll have time to talk to you about what builds are and all of those things. I'm sure Ilya's covering that in other courses. And now this is the mutation, right? So this is the codon usage, right? So this A is becoming a T. Of course, you also want to make sure the directionality of the, minus strand or plus strand, which means you have two strands, right? So it has one strand goes upstream, one strand goes the other direction. So you have to make sure which strand you're talking about. This is the codon. So what has happened is this one nucleotide change has changed the amino acid from E to V. There we go, causing a disease. So this is called anytime an amino acid actually is changed because of the result of a SNP, it is actually called a missense variant. Missense means that there is actually a change in the underlying amino acid that changes the protein and it changes the structure. So now this is another view. So this is the SNP. So you can see there are so many of these SNPs, right? So this is the position, and you have those two strands, right? One, the strand and the strand. And you actually can look at the SNP, and you can do lots of things from here, which I'm not going to go to today because it's beyond the scope for today. But then you can go, if any of you is working in a lab, you actually want to create a primer for and clone this particular region. You want to get the DNA sequence of just that particular region, or you want to do genotyping. You just want to look at just one polymorphism. Then you can actually use all of these things. And of course, a lot of different confusing things happen here. So I'm not going to add anything more because it could lead us somewhere else. And now the other database I want you to, and the other big projects that you can look at is actually Nomad, and also where it has all the sequences of exome sequences from population studies. You have Exact. You have all of these different population studies where you can see the frequency of this particular polymorphisms and all of those things. Now, I just want to quickly go to ClinVar. I love this database. Again, what ClinVar is, to answer Kylie's question is, if you go to ClinVar, and if, of course, you want to make sure that there is another database called ClinGen, that's one other day. So this is where it is telling you that this particular polymorphisms, all of these polymorphisms are clinically pathogenic, which means it is actually not good for you, right? So it is actually causing detrimental problems to the gene. That's what this means. It means pathogenic means that this particular SNP is not good. So it is actually pathogenic. So now we have begun to use all of this, whether the SNP is pathogenic or a SNP is benign or a SNP is, there are different things that they have actually come up with. So this is another example. So these are the two databases that in your own free time can actually, you can look at DBSNP, and actually you can also look at ClinVar, right? So these are two different databases. Now, very quickly, if I just go here, NCBI has actually a huge number of clean world, Muyiba, I don't know what you mean. Right, so there are lots and lots of, oh, I see, okay. Good. So there are lots and lots of, different databases here at NCBI. There are more than 100. And so if you just click here, and it's going to give you a lot of information. Now, remember, they've just incorporated a new database for just COVID literature, right? There are like 30,000 literature. If you just want to read only COVID papers, and it is, yeah, and then you actually you can do BLAST here. And I think Ilya is going to cover this or next week. So this is another database. Now, for literature, you can go all of to these databases. For genes, you can go through all of these databases. Each one we can create, hours of training for each one of this. I actually worked here, Consortium database. And I also worked in another database called Clusters of Orthologous Groups. And now this is the database we went to. For genetics, you have all of these different databases, right? Each one, when I say database, each one has millions of data within it, gigabytes of terabytes of data, or petabytes of data in it. Each one of this is a resource for data, right? Causing all this big data, right? And these are all the different genomes. SRA is a very good place to get all your sequences, which I am sure all of you have used because I know Ilia and the Pine Biotech platform actually goes there. Several of these databases you probably already know by using all the courses in Pine. And so all of these different databases. So we won't have time to go through all of these different databases. If we have time, we can get back. But for now, I think I just want to let you know that going to NCBI, clicking on some things, you can actually get lost. Now, one more database I want to take you to. If you go back to the hemoglobin page, are you all here? Hopefully I'm not lost a lot of you. Hopefully all of you are still following with me. I'm just going to give you a few seconds. Yes, good, Rahman. Thank you. Yeah, okay, good. Lots of you are following. Fantastic. So now let's click here, Ensemble. It's going to take a little bit of time. So here it is. This is my favorite place, right? So Ensemble. And again, I want to be very careful. If I start clicking here, we'll get completely lost, right? So it has a lot of different things in it. So what this gives you, what I love about this resource is it gives you population genetics, right? Now this SNP, right? That is actually, where do I click? You click on right here. Okay. Okay, so now I'm doing it again. So are you all here under, oh, I'm sorry. I actually should click here under sickle cell anemia. And there is something that you will see here, Ensemble. Okay, if you click there, did you all get here? Got it? Okay, Suparna, did you get it? Okay, so now this has lots and lots of information. So what I love about this is I use a lot of these different things. I like population genetics because what it tells you is that this SNP, because I told you, right, each of us are very different, right? So this is the allele, which is the ancestral allele. What does the ancestral allele mean? It just means that this is what is found in all populations, which is the ancestor. The ancestor at this position was a T, right? This is what it found. And minor allele frequency is an A, which means there are some people who have a minor allele, they can actually have A as well, right? And then of course, T means A, and T means A means T. And so this is a minor allele frequency. And these are also our other databases, right? So this is the SNP, and these are all the different databases that you can go to for clinical significance, which we won't be doing today. And I love this one. Just quickly, I am just going to click on population genetics here. And what you actually see is it actually tells you for all different populations, right? All populations together. This is all based on sequencing data. And all the population, 97% of the people actually have a T in that position. 3% actually have a A, which means A is tolerated for some populations, right? So in African American population, 90% of them have a T, 10% of them have a A. And then for American population, you see that 99% of the American population actually has a T. And then for East Asian, you have 100% of them have only a T in that position. Europeans and South Asians, right? All of this population has only a T in that position. So what does it mean? If this position in an East Asian or a European or South Asian population, if this becomes a C, what does that mean? It just means it's not going to be tolerated, right? Because evolutionarily, it's only T. In everybody, it's only a T. So if this is something else, maybe it's going to cause a problem, right? That's what this actually means. Of course, there are lots of different resources and frequency tables and all that. I don't want to go there because we might confuse you and all that, because it's just, I just was very, anxiously waiting to show you this resource because this is actually very, very good, right? So now I am going to get back to UniProt. So we went through DB SNP database. We went to Ensemble. We just went to ClinVar. And what else? So now I'm going to actually switch gears. I'm on sickle cell, but